All right, good afternoon, everyone. Welcome back. For this week, 2 sessions, we're going to talk about host defenses against virus infections. So this is basically a very fast immunology course. So if you've taken others, it's good. But we'll talk about just what you need to understand immune responses and vaccines and disease and so forth. So today we'll talk about innate and intrinsic defenses. So here's my view of immunity. A couple of barriers to virus infection. We start with anatomical and chemical barriers, things that are always present like mucus, which we've talked about, saliva, stomach acid, tears, your skin, an outer layer of defense, scabs, and defensins. These are antimicrobial peptides produced by your skin microbiome. Very cool. A lot of them are antiviral. And then if viruses get past that, which they can do, of course, they encounter intrinsic defenses. So these are defenses that are immediately available to counter infection. They include interferons, autophagy, apoptosis, microRNA, CRISPRs in bacteria and archaea. And the viruses that get past those then encounter the innate immune system, which is engaged within minutes to hours after infection. And that includes natural killer cells, NK cells, complement, antigen-presenting cells like dendritic cells, and macrophages, neutrophils, and cytokines. And that takes care of most infections, up to the innate immune defenses. In fact, if you are the biggest risk factor for severe COVID, as I told you before, is age. And after that, the second biggest risk factor is making antibodies against your own interferons. So that's a big part of the innate immune system. And so they're essential. And then some viruses get past innate immunity, and they encounter acquired immunity in the form of T cells and B cells. And we'll talk about that next time. So today we'll talk about intrinsic and innate defenses. So just to give you a clear picture of the distinction between the two. And remember, this is a human distinction that we make to try and understand and teach. Intrinsic defenses are always present in the uninfected cell. They include apoptosis, autophagy, RNA silencing, antiviral proteins, as you'll see today. And then the innate immune system is induced by infection. And that's not the case for intrinsic, right? The innate immune system is induced by infection. And then the adaptive immune system, which we'll talk about next time, is tailored to the pathogen. Now, there's some evidence that, in fact, innate responses are somewhat tailored, which is just a new emerging field of research. But for now, we'll say mainly the adaptive immune system is tailored to the pathogen. So let's look at a couple of examples of intrinsic defenses. One of them is RNA interference, which is a big defense in plant and invertebrate cells. Whether it's a big defense in mammals is not yet clear. There's some controversy in the field. This is a nucleic acid-based defense, right? And that's because plants and invertebrates don't have antibodies. So maybe that's why they rely on it more than do mammals. Anyway, it's in the form of RNA interference, which is a system inside the cell that takes incoming RNAs, say a viral RNA, and degrades them using a series of host proteins, and then uses those RNAs that are produced to target the viral RNA. So here's some incoming viral double-stranded RNA. It could be viral RNA in the particle, or it could be generated during infection. It is cut by an enzyme called dicer that cuts it into short pieces. Then one of those 2 strands becomes part of an RNA-induced silencing complex, or RISC. And then this is the siRNA, which is now complementary to the viral RNA, at least to one strand, so it can bind to it and induce its degradation. So the RISC has enzymes within it that will induce degradation. So it's a defense against incoming nucleic acids. And as I said, you can find this in some parts of mammalian development, but it's not clear what kind of a role it plays. It certainly is a regulatory role. Another interesting set of proteins are the APOBECs. Apolipoprotein B, mRNA editing, catalytic polypeptides. How many times fast can you say that? Really fast. APOBEC 3. This is it has other functions in the cell, but it is an antagonist for HIV-1 in particular. And the way it works is that it deaminates DNA. So here we have an HIV virus infecting a cell, and of course reverse transcription is occurring, converting the RNA into DNA. And the single-stranded DNA can be deaminated by APOBEC that is contained within the virus particle. And when you deaminate, you convert a C to a U, and then in the making of the other strand, you now have a base change. And so it introduces mutations throughout the genome. So it's a deaminase that introduces mutations throughout the genome. These proteins, by the way, have other roles in the cell, and they're thought to be dysregulated in some cancers as well. This gene, by the way, is present in many copies in different animals. So in humans and chimps, it's repeated many times. Each of these colored arrows are a duplicated APOBEC gene. And you can see they're duplicated in other species. And they're most duplicated in bats, which encounter lots of viruses. And so this may be part of the defense of bats against viruses. Anyway, the way the protein works, it's made in a cell. All right, so here is APOBEC 3G, that red oval there. And normally that would be incorporated into the virus particle during budding. So here's APOBEC 3. They're all different letters after the names, but here's APOBEC 3GF. It's being incorporated into the HIV particle, and then it will cause deamination of the DNA during reverse transcription. So why do we have HIV at all if this protein works? The answer is there's an antagonist encoded in the virus genome in the form of a protein called VIF. You can see this little blue square right there is VIF. It's a virus protein which basically it binds APOBEC, and it also binds a protein complex that leads to ubiquitination of APOBEC. So ubiquitination is the addition of small residues here. You see these, they're not nucleotides, they are a small peptide. And that targets proteins for degradation. When a protein has ubiquitin on it, one of the things that can happen is it gets degraded by the proteasome, which is shown there. So essentially, VIF makes APOBEC get degraded in infected cells. Yeah. So the question is, wouldn't it increase the mutation frequency? Yes, it would, but in this case, it mutates so much that it loses infectivity, right? If you have a certain, if you have every C in the genome mutated, that is not compatible with infectivity. So there's always a balance between mutation and viability. All right, so that's an antagonist of APOBEC, and that's why HIV exists, because it has this protein that degrades APOBECs. And you're going to see for all of these defenses, from today through Wednesday, there's always a viral counter defense to whatever it is. Yes. Okay, the question is VIF a target for antiviral therapy of HIV-1? There are no VIF antagonists that have been licensed. I'm sure people have tried. It's a hard target, right? It doesn't have an enzymatic activity. Proteins with enzymatic activity are the easiest to target with an antiviral because you don't have much of the enzyme. It's catalytic, so you don't need a lot of inhibitor. This is not enzymatic. It binds, so somehow you have to interfere with binding. That's very hard to do. So that's probably why we don't have any. Right. But in theory, it could be a target, yeah. Okay, so that's APOBEC and VIFs. Then we have epigenetic silencing. And this relates to something I told you earlier, and that is when viral DNA enters the nucleus, it gets wrapped around chromatin. We call that chromatinization. It gets wrapped around nucleosomes and silenced. And of course, viruses cannot have their DNA silenced, otherwise they would not replicate. But it's a kind of defense of the host cell to wrap the DNA around a nucleosome, and it will be silenced. And remember, if you modify the proteins in the histones, the chromatin, you can acetylate it or methylate it. You can open up the chromatin and get transcription. So the silencing is because the DNA is compacted by wrapping it around nucleosomes, and it can't be transcribed. So you can modify the histones to open it up. Viruses encode antagonists of epigenetic silencing. In particular, we have here human cytomegalovirus, Epstein-Barr virus, two herpes viruses, and adenovirus. These encode proteins that antagonize silencing. Now, when the viral DNA comes in the nucleus, it is not just wrapped around histones, it's shunted to what are called PML bodies, which are basically accumulations of DNA that have been silenced. And so some virus genomes some viral proteins impact the formation of PML bodies. For example, the EBV and adenovirus proteins block formation of PML bodies, so they block silencing. There is a protein in human cytomegalovirus that causes degradation of DAX, which is a protein needed for histone deacetylation. So deacetylation of histones would compact the chromatin and cause it to be silenced. All right, and so this virus encodes an inhibitor of that process to keep it acetylated and open. And then finally, remember, retroviral DNA comes in the nucleus and it integrates. If there's any DNA that remains unintegrated, it's silenced. But once it's integrated, it is not silenced anymore because it's in the genome. It's next to some gene that's active. So integration is actually an anti-silencing strategy, if you will. By going in the genome, it assures that it's not silenced. And of course, we have apoptosis, programmed cell death, right? Which happens normally in us to get rid of cells that we don't need. We have a normal cell, and a program of apoptosis is induced by a variety of ligands and receptors, and that ends up in the cell basically dying and being taken up by other cells. This is often a response to virus infection because it's a way of preventing the virus from reproducing. If the cell is dead, the virus can't reproduce in it. You sacrifice a few cells, but maybe you save the organism. So at the top is a diagram of apoptosis, and there at the end is a macrophage picking up apoptotic bodies. There's an electron micrograph there showing you those apoptotic bodies blebbing off from the cell. Now this is important in immune responses. Apoptosis is monitored by sentinel cells like macrophages and dendritic cells and NK cells. They will take up these apoptotic bodies. They will take them up by phagocytosis and go to a lymph node and present them to a T cell, who will then decide if there are any foreign proteins in that infected cell. So it's a part of the way of responding to a virus infection. We'll talk about that next time in some detail. But apoptosis is another intrinsic defense against infection. And it's highly regulated, as you may imagine. And this slide is just meant to give you the overview of that. I don't want you to know any of these details, except that there are a couple of receptors on the cell surface which, when engaged by ligands, cause the cell to undergo apoptosis. Now you have the FAS receptor, the trail receptor, and the tumor necrosis factor receptor. When they engage their ligands, FAS ligand, trail, or TNF, that induces apoptosis in the cell, and there are a variety of signaling pathways that are involved in that. And you have caspases, which are proteases that can be activated and eventually induce apoptosis. Now, viruses interfere at multiple levels with apoptosis, and those are shown by the red arrows, the red lines, I mean. So these viruses block interaction of receptor with ligand. These proteins of these viruses block activation of the receptor and downstream events. And you can see viruses are interfering at different places. But interestingly, some viruses stimulate apoptosis, as you can see here. And this is presumably to help viruses leave the cell at some point. So the stimulation doesn't happen right away, of course, because then you'd interfere with reproduction. But it does happen later in the infectious cycle to facilitate viruses leaving the cell. There's also a form of apoptosis that is stimulated by mitochondrial damage. So if mitochondria get damaged, the cell, this is very bad for a cell not to have mitochondria. The cell might as well die, and it does. So the breakage of mitochondria leads to release of cytochrome C, which is then recognized by a caspase and turns on what's called the intrinsic apoptosis pathway because it happens within the cell. But this is, again, an intrinsic defense in general against virus infection. And finally, there's an ancient defense, an intrinsic defense known as CRISPR. So this CRISPR system, clustered regularly interspaced short palindromic repeats, this has evolved in bacteria and archaea to defend them against viruses and probably plasmid DNAs coming into the cell. And of course, the beauty of it is that it has been repurposed by humans for gene editing. But it wasn't originally made for gene editing. It's a defense against DNA invasion. So here you have a bacteriophage infecting a cell, a bacterial cell. The DNA comes in, and the CRISPR nucleases will chop it up and integrate pieces of that genome into the host cell DNA. And then those bacteria that survive, they will transcribe that array of integrated host DNAs into what's called CRISPR RNAs. And then the next time a phage infects that cell, the CRISPR RNA will direct a nuclease to the viral DNA and cut it, and therefore defending against infection. So it's a really nice defense against infection. It's in 90% of archaea, 50% of bacteria, it's been found. By sequencing the bacterial genome, you can find all of these integrated pieces of viruses. So you can figure out what viruses infected that bacteria. So if you do environmental sampling for viruses, let's say, and you find sequences of all these different viruses, you have no idea what their host is, you can search in the genome, the cell genome, for a CRISPR array that would match the virus to assign the host. That's a commonly done thing. It's done bioinformatically, of course, but it's a beautiful way to somehow assign the host of certain viruses. So our first question is, intrinsic defenses are always present. Which of the following are included? Antibodies, T cells, epigenetic silencing, skin, or mucus. So what did we do after all that? So epigenetic silencing is the answer. It's an intrinsic defense, always present. Antibodies is not intrinsic, or T cells. We're going to talk about those on Wednesday. Skin is a, what do they call it? It's in this slide. It is an anatomical and chemical barrier. And mucus is also an anatomical and chemical barrier. So, okay. I thought that would be an easy one. See, that's anatomical and chemical and intrinsic. Acquired immunity is the T cells and B cells. All right, let's talk about the innate immune system, which is that third brick wall where it's activated minutes to hours after infection. And it's comprised of cytokines, sentinel cells, and complement. Cytokines, sentinel cells, complement. The sentinel cells are dendritic cells, macrophages, and NK cells. We've already encountered those a bit because they can sample the apoptotic bodies to see if there's something foreign there, like a virus. And the neat thing about the innate response is it communicates with the adaptive response. If the innate response cannot handle an infection, if virus levels get too high, then the innate response hands it off to the adaptive response, and that takes over. And we'll see how that works today and next time. So the innate system has been known for many years, but the key question always has been, how does an innate system recognize foreign and not self? And a key discovery, well, people always thought there must be some receptors that recognize parts of viruses as foreign. And the key event was in 1980, two scientists were studying Drosophila. And they were making mutations in the fly and trying to find genes that regulated development. So this is all done very routinely now, but back then it was pretty new. And they found one gene called the Toll gene, because it means das war ja toll in German, which is like crazy, far out, right? And so they called it the Toll gene. And they got the Nobel Prize for discovering that in 1995. But it was a gene playing a role in Drosophila development. But it turned out, as found later, that it played some role in immunity of flies to microbes, and then later on that it plays a role in innate immunity in mammals. So these are the toll-like receptors, which are part of the innate immune system, so originally discovered in Drosophila. So you remember we had a presidential candidate once who said, why are we studying flies and worms? Do you remember who that was? You're too young to know that. It was the governor of Alaska who said she could see Russia from Alaska, so she knew all about the politics. Do you remember? Yeah, I think that was her name. She said, why are we spending money on flies and worms like C. elegans and Drosophila? So, I mean, this is one of many examples. You can find out really neat and important stuff, because now this has led to a whole new area of research of receptors that distinguish foreign from self. We call these pattern recognition receptors, or PRR. There are four different categories shown here. There are C-type lectin receptors that recognize glycans, basically, in pathogens. There are toll-like receptors, as we just found in Drosophila. These are transmembrane proteins that recognize foreign proteins and nucleic acids. There are nucleotide binding receptors in the cytosol, and there are RIG-I-like receptors in the cytosol as well. And these all recognize something that is different about a pathogen. And they can be recognized in them at the cell surface, as you can see here, for the plasma membrane proteins, or within an endosome, or within the cytoplasm. And let's take a look at some of these. So the recognition of something foreign, that foreign entity is called a pathogen-associated molecular pattern. And one of them is nucleic acids, right? And that's figuring prominently on this slide. But there are also viral proteins that are recognized. For example, toll-like receptor 2 and 4 can recognize viral proteins on the plasma membrane. And here we see viral RNAs being recognized. So toll-like receptor 3, 7, 8, and 9 reside in the endosome, and they can recognize different motifs like double-stranded RNA, single-stranded RNA, etc. And the reason they're recognized as foreign, because there shouldn't be any double-stranded RNA in a cell, for example. There shouldn't be any single-stranded RNA in the cell. If there is, they're usually viral. So if they're present, and the reason they're in an endosome is because that's where many viruses are taken into the cell. And also on the right is a system for sensing cytoplasmic RNA, because not all viruses are going to come through an endosome, right? Some of them come in at the cell surface. And so you can see that double-stranded RNAs and 5' phosphorylated RNAs and long double-stranded RNA, these are all motifs not found in the host cell. That's the key here, that these are recognized because you don't see them in the host cell. And in all cases, the recognition leads to signaling pathways that turn on the synthesis of interferons, for example. So here on the left, this endosomal sensing system leads to the entry of transcription proteins into the nucleus, and they turn on the expression of interferons, which we'll talk about in a moment. The same with these cytoplasmic sensors, which are called RIG-I and MDA5. They lead to the entry of transcription proteins, IRF3, 7, NF-kappa B, into the nucleus, and they turn on the synthesis of interferons. And what interferons do, we'll look at in a moment. But this is the very first step, where there's something in the cell, a protein or a nucleic acid, that is sensed as foreign, and the cell begins to make interferons, which in turn can have some antiviral effect. DNA is also sensed in particular, in this case, is a system for sensing DNA in the cytoplasm of the cell. So DNA should not be in the cytoplasm, and if it is, it's assumed to be a virus infection, or at least foreign, and it shouldn't be present there. And there is an enzyme in the cell called cGAS, which will recognize, here it's recognizing double-stranded DNA. This stands for cyclic GA synthase. So it takes a molecule of ATP and a molecule of GTP and makes a cyclic version of them, cyclic GAMP, and that's shown here on the right. That's cyclic GAMP. That molecule is made when cGAS senses double-stranded DNA in the cytoplasm. And then the cGAMP binds to a ER protein called STING, which stands for stimulator of interferon genes. People have made jokes about STING being the police of DNA sensing. You get it? I don't know. If you're too young to know police, right? I just don't know. STING, that's STING. But you know, they probably made up STING by using certain words. Anyway, STING binds this and then the cGAMP, and then it translocates to the Golgi, where it then activates some protein kinases like TBK, and that phosphorylates transcription proteins like IRF3, which go in the nucleus and turn on interferons and other cytokines that cause inflammation that we'll talk about in a moment. So again, sensing DNA turns on a signal. There are also nuclear sensors of DNA which respond to viruses, viral DNA. And of course, every virus genome has to encode some kind of antagonist of this sensing. Otherwise, there wouldn't be any viruses around. These are very powerful systems. So we have, all in red here, are antagonists of these various systems that we've talked about. For example, the cytosolic sensing of RNA by MDA5 and RIG-I. It's antagonized by multiple viral proteins at different steps. For example, this KSHV, Kaposi's sarcoma herpes virus protein, blocks ubiquitination of RIG-I, which is needed for its activity. The other proteins block the signaling pathways downstream of the sensing. And the same goes for sensing of DNA by STING as well. It's interfered with by multiple viral proteins. So you have to have an antagonist for these very powerful sensing procedures. That's a theme. Everything that we talk about, every cellular defense that we talk about is going to have a viral counter defense. Okay, next question is, which of the following allow the innate immune system to distinguish microbes from self? Cytoplasmic helicases and toll-like receptors, antibodies, apoptosis, APOBEC, all of the above. You have a chance to redeem yourselves from the previous performance. Let's see how we did. Cytoplasmic helicases and toll-like receptors are how the innate immune system distinguishes microbes from self. That's correct. So antibodies, they don't. They are tailored to the pathogen, but they're not about early recognition, right? They're not about the innate immune system. That's basically why they're wrong. They're the adaptive immune system. And apoptosis and APOBEC are the intrinsic, which is before innate. And I'm sorry that it's all about terminology, but just to get you to think about it in a way that we think is useful, that's what that's about. So we've mentioned interferons, right? Interferons are turned on. The synthesis of interferons is turned on when the innate immune system detects something foreign. So what is an interferon? Great name, by the way, isn't it? Interferon. And it was coined back in 1957, where two investigators were exposing they would infect chicken cells with influenza virus, and then they would take the supernatant and put it on another cell, and that would make that cell resistant to virus infection. And they said it was interfering with infection, so they called it interferon. And basically, these are proteins produced by both virus-infected cells and by Sentinel cells in response to viral proteins, viral nucleic acids, and viral proteins and nucleic acids. So here, for example, is a sheet of epithelium somewhere in our body. It is being infected by viruses, and those infected cells detect virus infection through the innate immune system using the receptors I just talked about. They produce cytokines, which are proteins including interferons and other proteins, as we'll see in a moment. The cytokines will attract immune cells to the infected area to try and clear the infection. And among those immune cells are dendritic cells and macrophages, which will be activated by the cytokines. They will pick up proteins from the dead and dying cells and bring them to the lymph node, which we'll talk about later. So there are three kinds of interferons that we want to talk about: type 1, type 2, and type 3, or alpha, betas, gamma, and lambda. So the interferons are soluble proteins, and they have receptors that are distinct for each one of them. So here on this picture, we have the three sets of ligands receptor. Interferon alpha is the purple oval shown binding to its receptor, and interferon gamma is there, the two purple circles, and then interferon lambda, the red oval, and each of them binding to a distinct receptor. And when these interferons bind the receptor, they activate it, and so it can phosphorylate downstream proteins and start a cascade of gene expression, as we will see. So the interferons are turned on by sensing foreign proteins and DNA by the innate immune system. So here is a virus-infected cell. There are two cells here. On the left, virus-infected cell. It's making single-stranded RNA or double-stranded RNA that's sensed by cytosolic sensors that turns on transcription of interferon genes in the nucleus. And those interferon genes give rise to mRNAs, which are translated, and the proteins are secreted. The interferons are secreted from the infected cell, and they can bind to receptors on other cells. And then those other cells will turn on the synthesis of what we call interferon-stimulated genes, or ISGs. And those ISGs are what have antiviral properties. So the interferon, in this case lambda, is binding its receptor. We have a series of events occurring downstream which lead to a transcriptional activator going in the nucleus, and that turns on the synthesis of interferon-stimulated genes. So this is in general what happens for each of those three interferons. They're turned on in infected cell, they bind receptors on other cells, and then they induce the antiviral proteins. They induce the synthesis of antiviral proteins. So these promoters that are responsive to interferon, so here interferon is binding a receptor, and eventually promoters are turned on downstream. Those promoters have elements of endogenous retroviruses in them. And so that's shown on this side. Endogenous retrovirus, LTRs, regulate the interferon response. Remember, the LTRs are present at either side of the provirus. Here's an endogenous retroviral provirus. It's got two LTRs, right? It drives production of retroviruses. But cells have taken those LTRs and used them for other things. So here is one LTR that's been translocated somewhere else in the genome by recombination, and it can drive the expression of a host gene. And these host genes include those interferon-responsive proteins that I just showed you in the previous slide. In other words, they will be activated by interferon binding to the cell surface receptor, and the transcription factors go in the nucleus. They will bind to elements in this repurposed LTR and turn on the synthesis of interferon-stimulated genes. And this kind of exaptation of the LTR is the right term for it, has happened in many different animal species, as you can see here on the right. And it's happened many, many millions of years ago. So these are ancient infections that led to establishment of these proviruses. And then with time, the LTRs are useful as promoters, so they're placed elsewhere in the genome. It's another way these retroviruses are useful, right? They give rise to a placenta, they give rise to blue eggs in chickens, and now they are giving rise to antiviral responses. So what about these interferon-stimulated genes? What are they? There are over 1,000 of them. And I'm not going to go over all of them with you. What's the point of that? I want to go over a few so you get a sense of how they work and the ones that we know about. One of them is called tetherin, or CD137. This is very cool. This is a protein induced by interferon, right? Its promoter is turned on in the nucleus by that interferon pathway. It goes and binds to membranes. And as viruses are trying to bud, it locks them to the cell surface or to the endosome, as you can see here. So this is tetherin, this dimer that's present in the membranes. It binds to the cell membrane and to the virus membrane and prevents the viruses from leaving the cell. And you can see on the right is an electron micrograph of an HIV-infected cell where the tetherin is preventing the viruses from leaving, and they're just stuck on the surface of the cell. So why do we have HIV if we have all these amazing antiviral proteins like APOBECs and tetherins? Because the viral genome encodes an antagonist. The VPU protein of HIV antagonizes tetherin. It makes it degraded, and so it never gets to hold the virus on the cell surface. But if you take VPU out of the virus genome, then the virus gets stuck at this point. But this is an interferon stimulated gene. Another one that I think is very interesting is called IFIT1. Interferon-induced protein with tricopeptide repeats number 1. This is a protein that binds RNAs lacking 2' O methylation. And so capped RNAs usually have this 2' O methylation. So here's a cap at the end, the 5' end of the RNA, and then the next two bases are 2' methylated, and IFIT recognizes that and binds to the 5' end. So here's a messenger RNA where IFIT is binding it, and then if IFIT binds, it can't be translated because the initiation complex, which depends on interacting with the cap, doesn't function. And so on the right is a structure of RNA and IFIT. So the IFIT is the protein with all the alpha helices, of course, and the RNA is the red inside of it. And there at the very top is the M7G, which is the cap on the 5' end. And on the right is a space-filling model where you can see there's a channel in the protein, in the IFIT protein, that the RNA fits in, and it locks itself very tightly on it, so it can't be translated. So this protein will basically stop the translation of any RNAs with a 2' O methylated cap, which includes host RNAs too. So that would make the cell sick, but you have to make a decision, what you're going to save. However, viruses get around this, right? How do they do? There are a couple of different ways here. First of all, what I just told you is slightly wrong. Let me go back. Cellular mRNAs have 2' O methylation. IFIT binds mRNAs that don't have those methyl groups there, all right? So how do viruses get around that? Well, influenza virus, its 5' end is stolen from the host cell, so that has 2' methyl groups on it. So that's how flu gets around IFIT1. The coronaviruses don't have a cap. They have a little protein stuck to the 5' end, VPG, so IFIT can't bind, can't inhibit translation. Some viral genomes encode their own 2' O methylase. Isn't that amazing? They can methylate their mRNAs so they're not recognized by IFIT. And some viruses steal host methylases to methylate their own caps. And finally, some viruses have an RNA structure at the 5' end that prevents the protein from binding because the double-stranded structure goes right up to the cap, so that it can't fit into the channel in IFIT. So many ways that viruses escape inhibition by IFIT. And I think this is the last one. This is another IFIT M3. This inhibits fusion. So IFIT M3 is this protein on the right here that has a transmembrane segment, that's the blue part, that's the phospholipid bilayer, right? So the protein sticks in there, but then it has an alpha helix and some lipids attached to it that make it insert into only one of the leaflets of the membrane. So the effect of that is it blocks fusion. So here in the bottom is fusion between a virus and host membrane. So you remember, you have fusion proteins that cause hairpinning, and eventually you want to have both leaflets fuse, but the presence of IFIT blocks fusion. It can't go through that final step because it's disturbing the membrane in some way because it's stuck in there in two different ways. And so IFITs will block fusion. IFIT M3 will block fusion at the plasma membrane. It will also block fusion from within endosomes. And again, there are viral antagonists of this, which is why viruses can still exist. All right, so those are some interferon-stimulated genes and how they work. Now, interferons induce lots of different interferon-stimulated proteins. Over 1,000 of them, I said. And basically, every cell on your body has some kind of interferon receptor. So if you have a virus infection, the interferons typically diffuse throughout you. So if you have a nasopharyngeal infection, you're going to make interferons locally, but they're going to get into the circulation and they're going to be in your brain and they're going to make you have a headache or make you feel lethargic, right? So interferons are dangerous, actually, because they can harm you. They cause fever, they cause chills, nausea, malaise. Whenever you have a virus infection, you feel pretty much the same thing at the onset. We call this flu-like symptoms, but they're not really just induced by flu. They're induced by any virus infection that makes interferon. Because every viral infection results in the production of interferon, and therefore, most virus infections result in these flu-like symptoms. And that's also a reason why if you treat people with interferon, they feel miserable. So for hepatitis C, we used to treat people with interferon for months. Can you imagine having flu-like symptoms for months? Most, you can. It's really nasty, I know. And so most patients who were taking interferon would stop because they couldn't handle the symptoms. So we don't do that anymore. Fortunately, we have better antivirals, but it is a problem. And you know, at the onset of COVID pandemic, people were thinking about using interferons as treatment, but it never really gained much traction. And it's partly why. Okay, the last question is, how do interferons limit viral replication? We have interferons directly inhibit viral translation. Interferons lyse virus particles. Interferons induce ISGs. Interferons damage cells, none of the above. How do interferons limit virus replication? All right, let's have a look here. A little mixed bag here, folks. The answer is interferons induce ISGs. That is how they limit virus replication. They do not directly do anything to a virus-infected cell except induce ISGs. So they don't inhibit viral translation on their own. IFIT1 would, but not interferon. Interferons don't lyse viral particles, and it's not none of the above because it's that they induce ISGs. So remember, this is a key point. Interferons are antiviral because they induce other proteins that have antiviral activity. They're not in themselves antiviral in any way. Let's talk about the sentinel cells that are part of this innate immune system. I mentioned them briefly before because they were picking up apoptotic bodies, like a dying cell. You know, a cell dying could be dying from natural processes, or it could be virus infected. So in all cases, the sentinel cells pick up these apoptotic blebs and they bring them into the cell, they process the proteins and bring them to lymph nodes. And so we have three sentinels: dendritic cells, macrophages, and NK cells. And they're always circulating through you. As I told you the other day, we make 1 billion NK cells a day per kilo of your body weight. It's a huge amount of cells, and I don't know about the others. But these are photographs of dendritic cells. They're called dendritic cells because they have dendrite-looking processes. Not that they have anything to do with the nervous system, but they just look that way. And on the right is an NK cell that is attacking a virus-infected cell, which we'll talk about in a moment. Now, dendritic cells, there are many different kinds of dendritic cells. As you can see, there are ones in the blood, there are ones in different tissues. But what they do is patrol organs and mucosal surfaces. So you can see in the intestine, that's a model of the intestine there with our epithelial cells. Just below the basement membrane, there are dendritic cells, these blue and purple cells. And the red are virus particles, of course. And the dendritic cells are taking up particles and pieces of dead cells to see if they're foreign. And how they do that, I'll tell you in a moment. But you can see even this one dendritic cell is sticking one of its dendrites through the junction between the cells and sampling the lumen of the intestine. Quite interesting. Also, their epithelial cells have dendritic cells that patrol them, the skin, the eye, the vagina. They have dendritic cells that, again, do the same thing. They're looking for a virus-infected cell. Now, the dendritic cell itself can't know if anything it picks up is foreign or not. That job is up to the T cells in the lymph node. So what dendritic cells do is they get into the lymph circulation. The lymph capillaries are very permeable. They're throughout your body, and the dendritic cells go into them, and eventually lymph capillaries go to a lymph node, which is shown here. And lymph nodes contain zones of B cells and T cells, and that is where the interrogation of the protein that the DC has picked up, or the macrophage, is done. The dendritic cells, in this case, present their findings to a T cell, and then the T cell decides whether it's foreign or not. And if the antigen is foreign, the T cell becomes activated, it proliferates, and leads to the production of T cell effector T cells and antibodies, as we'll see next time. But there is a very cool picture on the right, a fluorescence image of a dendritic cell in green, which you can see by its morphology. It looks like a dendritic cell, but you can stain it with for other markers to prove it's a dendritic cell. And then the blue cells are T cells. This is in a lymph node. And you can see the dendritic cell is snuggling right up to the T cell and presenting what it's found in the periphery. And so that's summarized here schematically. So we have virus infection of an epithelial cell sheet. The virus has infected the sheet, and now the cells have sensed virus infection. They're making cytokines, and the cytokines attract the dendritic cells. When they're first going to the infected cell area, they're what's called immature. And then the cytokines and viral proteins or nucleic acids released from infected cells also will be recognized by the dendritic cell. They activate it, and it becomes more looking like a dendritic cell, as you can see here. That then goes into the lymph node, where it presents what it has to the T cells. And here's what happens in some detail. There on the left is an immature dendritic cell. Dendritic cells have everything. They have all the sensors of foreign products. They have toll-like receptors, cytosolic receptors. They've got it all. They also have MHC class 2, and they have receptors for cytokines. They can make interferon. And so when viruses or virus proteins are taken up by the dendritic cell, they can be sensed, and the dendritic cell can make interferons, which will be important later, but those interferons can also assist at clearing the infection from the area where the dendritic cell has gone. So the DC takes up viruses, virus proteins, dead and dying cells produced by apoptosis. They're also turned on by cytokines produced by the infected cells. And all of this interaction causes them to mature. So what does that mean? Well, the morphology of the DC changes, as you can see, but now MHC2 is on the cell surface, the plasma membrane. And MHC2 is the cell surface protein that's going to present peptides that the DC has picked up from virus proteins, dead and dying cells, and so forth. These are processed in the DC and chopped into small peptides and loaded onto the MHC2, and eventually those MHC2 go on the surface of the DC. So when the DC, which is now a mature DC, goes in the lymph node, it will present that peptide to a naive T cell via the T cell receptor. So that little orange rectangle is a peptide. It could be self, it could be non-self. But in theory, during development, during fetal development, all the self T cells are eliminated. It's like elementary school for T cells. If you react with self, you're kicked out. It's not so bad as in real life, because these T cells are forced to apoptose and die, which would be harsh in real life, right? So any self-reactive T cells that recognize self-peptides are supposedly gone, but in reality, we have lots of diseases where they're not gone, and those are autoimmune diseases. So if you recognize a peptide, if there is some T cell in you, and these MHC are polymorphic, so are the T cell receptors. So there's one that matches a peptide somewhere in you. If one matches a peptide, the assumption is it's foreign. That T cell will become activated and then go on to do its immune functions. So the way that the T cell recognizes the peptide as foreign is through the T cell receptor presented by MHC2. The dendritic cell is also producing cytokines and interleukins and so forth that help mature the T cell as well. And so that's what's going on in the lymph node when this happens all the time, even when you're not infected. But if you're infected, some viral proteins are going to be taken up by those dendritic cells, and this process will determine whether the innate immune system activates the adaptive system. We also have NK cells, which will kill virus-infected cells. Now, these are also taking up antigens. They're antigen-presenting cells, much like APCs, but they also have another function, which is to kill cells that don't have MHC on the surface. So many virus infections down-modulate MHC molecules, right? So all cells have some kind of MHC, which is involved in presenting antigens. So here's a target cell where there's no MHC, and that's not normal, because all of our cells should have MHC. When there's no MHC, the NK cell is assuming it's infected, and the virus has downregulated it, and it kills the cell. So this is called absence of self-killing, because MHC defines self, right? It's how you know your cells are yours, by your MHC molecules. But guess what? Some viruses can put a mimic of MHC on the surface, so the NK cell thinks it's not infected and thereby gets around it. On the bottom is shown a cell that's uninfected because it has an MHC, normally functioning MHC, and that prevents the NK cell from killing it. And there are many modulators of NK cell activity. And finally, I want to tell you about complement, which is a series of cell proteins. This is considered really a link between the innate and adaptive responses. A series of cell proteins that can recognize different molecular patterns, which will then lead to production of cytokines and lysis of membranes, or enhancing antibody binding, as we'll see next time. So here we have three different ways that complement proteins are doing this. All these proteins with the C names, these are all complement proteins. They're all present in your blood all the time, and they're ready to be activated by infection. For example, if there's a virus or a bacteria with a particular carbohydrate on it, that can be recognized by specific binding proteins and activate the complement pathway, leading to the production of cytokines, and even a production of a membrane attack complex, which is a big protein complex that could poke holes in membranes, and presumably would poke holes in the bacteria or even the viruses. Here's an example on the left of when antibodies are bound to a microbe. one of the components of complement C1 will recognize those antibodies bound, and they activate this pathway, leading to, again, inflammation or membrane attack and so forth. So that's the complement pathway. And yes, there are modulators of complement because it's a very effective pathway. So all of this that I've been talking about so far is leading to what we call inflammation. So the production of interferons and cytokines and even chemokines by this response system causes what we call inflammation. And you've all had inflammation, whether you're infected or not. If you hurt yourself, when you cut yourself, you get inflammation. You get swelling and redness. When you injure a muscle, you get inflammation. When you get infection, you get inflammation because the cells are producing cytokines and chemokines. And that diagram shows you all the different causes of inflammation, except virus infection or infection period is not included. I don't know why they wouldn't include it, but all these diseases are associated with inflammation. So what is inflammation? Redness, pain, heat, and swelling. These are the four classic signs originally described by this Celsus, a Roman medical encyclopedist in the 1st century AD. And they're called in Latin rubor, dolor, calor, and tumor, right? Redness, pain, heat, and swelling, all caused by the changes induced by cytokine and chemokine production. Increased blood flow to an infected area to try and get more immune cells, increased capillary permeability so that cells can cross the capillaries, and that is causing fluid leakage and pressure and pain, influx of phagocytic cells, also leading to swelling and tissue damage. So all those things are resulting in redness, pain, heat, and swelling. So this is what happens during an infection, and sometimes you can see it. So if you have an upper respiratory infection, all those signs and symptoms you get are, or the symptoms you feel are a consequence of inflammation. We recognize three different kinds of cytokines. There are pro-inflammatory cytokines, which cause all these signs of inflammation, like IL-6, TNF, IL-1, IL-12. Those are famous pro-inflammatory cytokines. And some viral diseases, a big component of severe disease is inflammation. Like the severe part of COVID after the viral phase is an inflammatory phase where lots of IL-6 and TNF are being made. So after a week of the viral phase, if your infection continues, you're in an inflammatory phase, and you can use immune suppressants to take care of that. You can use dexamethasone, which would be a broadly inhibitory suppressor, or you can use antibodies to IL-6, which is one of these pro-inflammatory cytokines. Then we also make anti-inflammatory cytokines, which reverse the action of the pro-inflammatory cytokines, right? There has to be a way of turning off the system, and that's what the anti-inflammatory is. And finally, chemokines that recruit immune cells. So chemokines would be produced by an infected cell that has sensed infection, and that recruits the antigen-presenting cells like the dendritic cells. And there on the left is a diagram of what's happening in general. We have a sheet of cells in your body somewhere. It's being infected. It releases cytokines and chemokines that attract cells, including neutrophils. So neutrophils are present in the blood, and they can be attracted to the infected area by chemokines. They have the unique property of being able to squeeze through the spaces between the endothelial cells of the blood vessels. And this multi-lobed nucleus, the reason it's in three pieces like that is so it can help squeeze the cells, squeeze through the blood vessel. Now these chemokines and cytokines are made to combat a local infection, right? Wherever in your body the infection is, they're made locally, but they get in your blood and they diffuse all over. And they enter your circulation and have global effects: sleepiness, lethargy, muscle pain, no appetite, nausea, all these flu-like symptoms. So even a local infection in your upper tract can have systemic effects, and it's because of these cytokines that are made that happens. Now, the cytokines themselves work by binding receptors on cell surfaces. And here on the left is an example of that, where we have a cytokine at the top binding its receptor. It initiates a signaling cascade, which eventually lets transcription proteins in the nucleus, and then you turn on genes that execute the cytokine's function, if you will. And an example of that is shown on the right, where we're looking at neutrophils in a blood vessel that are going to sense infection in a local tissue, and they're going to get through the blood vessel. And what happens is the endothelium produces certain membrane proteins induced by cytokines. And those include proteins shown here, like Ig superfamily CAMs and selectins that will bind the neutrophil, and other chemokines like CXCL8 will bind receptors on neutrophil and upregulate integrins. The result is the neutrophil attaches to the blood vessel wall. This is an endothelium. You know, the blood flow is really fast, so you have to have really good receptor ligand interactions to stop these cells, and then eventually they will squeeze through. So that's just one of many outcomes of cytokine production. And of course, viruses counter cytokines. Of course, otherwise they would be eliminated. Here are each of these columns show a different way that viruses counter cytokines. They can inhibit the production of cytokines. They can interfere with how they work. For example, some viral genomes encode homologs of cytokines that will bind cytokine receptors but don't signal. So you don't get the downstream events that would be caused by the cytokine. And some genomes encode soluble cytokine receptors that are secreted from the cell, and they bind up all the cytokines so they can't act. And then finally, some viral proteins work in the cell by interfering with signaling during cytokines binding to the receptors. Now, inflammation sounds bad, and it is in a way because it hurts, but it also stimulates potent immune responses. So what we have found is that cytopathic viruses that kill cells cause inflammation, and inflammation links the innate and the adaptive responses, so you get better antibody responses. So cytopathic viruses typically cause good inflammation, and viruses that are cytopathic, like adenoviruses, herpes viruses, pox viruses, they often encode antagonists of innate immunity so that they don't get this inflammation. There are also viruses that don't encode, that don't kill the cell, and they don't make good inflammation, and therefore you don't get a good immune response. So these viruses we call non-cytopathic. Cells are not damaged, and no bits of dead and dying cells are released for the sentinels to pick up. Therefore, you have a poor adaptive response, and these viruses are typically persistent. They infect you for many, many years because they aren't cleared initially. And again, it's all because we didn't have good inflammation during the innate response, which, as you'll see next time, leads to a really good adaptive response. So the lesson is that inflammation really reflects the communication between the innate and adaptive responses. If you have good inflammation, you have a good adaptive response. If you have poor inflammation, you don't make a good response, and you probably won't be able to clear the infection. And this is one reason we use adjuvants in vaccines. So vaccines that do not replicate, protein vaccines, we use adjuvants to stimulate inflammation and get you a better immune response. Some of the best vaccines have an adjuvant. In fact, the mRNA vaccines are encased in a lipid nanoparticle that in itself is an adjuvant that stimulates inflammation. Now, even as I told you before, not all inflammation is caused by infection, right? If you scratch yourself, you will get inflammation as part of the repair process. And in fact, one vaccine, the smallpox vaccine, which we don't use any longer except for certain populations, involved scratching your skin. And the act of scratching caused inflammation that made a better immune response to the vaccine. So the smallpox vaccine was delivered in this bifurcated needle shown here on the left. A drop of vaccine put in the needle, and then on your muscle up here, they would scrape it in. and then you'd get this very nice scar, which I have and probably few of you have because we stopped immunizing in 1979. Although if you're in the military, you would be immunized. And that scratching causes a break in the skin. It causes the production of cytokines and chemokines, attacks immune cells. And if you don't do that, if you just inject the vaccine, you don't get anywhere near as good an immune response. So this inflammation is an important component of the efficacy of that vaccine. Most of our other vaccines are injected, and that doesn't cause very much inflammation on its own. So the inflammation comes from the virus reproducing or from an adjuvant. So the last thing I want to leave you with is that I hope you got from this discussion that all viruses have to encode at least one regulator, all the defenses that we've talked about today. That includes sensing, interferon production, signal transduction, cytokines, chemokines, NK cells, DCs, complement. There's always an antagonist because these measures are so effective. They could, in theory, eliminate virus infections, but the viruses have evolved, or evolution has provided viruses with countermeasures. So here's a summary of what we did and what we're going to do next time. We have an infected cell sheet. The cells sense infection via the innate immune system. They produce cytokines. The cytokines recruit antigen-presenting cells like dendritic cells. The dendritic cells become activated. They pick up pieces of viruses or dead and dying cells. They deliver it to the lymph node, and there they talk to the T cell. And what happens there is the subject of Wednesday, where we'll talk about how antibodies and T cells are made and what they do.